When we're considering the hydrophobic side chains of the amino acids, it's important to commit the structures to memory. What's equally important, though, is to develop a sense of the characteristics of those side chains so that we can place them in categories and make use of those categories as we encounter real situations with real proteins, real contexts. What are the amino acid sizes? What are their relative hydrophobicities? Are they cyclic? Are they heterocyclic? Do they have shared pi electron systems? Are they aromatic? Are they branched? At what carbons are they branched? These are some of the categories that we could use to organize our thinking about amino acid side chains. If we consider phenylalanine first, we simply see a benzene functional group attached to the backbone of the polypeptide through the beta carbon. We note that there's a three-letter code, PHE, for phenylalanine, and its single letter code, F. Tyrosine is a slightly more complex side chain. It's got that same benzene ring, but in addition, at the para position, it has a hydroxyl group. We know that this is an ionizable group with a pKa of around 10. It's not ionized at physiological pH, but that hydroxyl group plays lots of important roles in various kinds of structural and catalytic examples. One important thing to think about, though, is that tyrosine is a side chain that, unlike phenylalanine, which is usually entirely buried, tyrosines are often partially exposed. The benzene ring is buried in the oily hydrophobic core, and the hydroxyl group is exposed. Tryptophan, the largest of the hydrophobic side chains, another aromatic side chain with a large shared pi system. Uh, it has a strong ultraviolet absorbance because of that shared pi system. We'll encounter that later. It also engages in pi cation interactions, which are worth mentioning just because they're so counterintuitive. The distribution of electrons in that heterocycle are such that there's some partial negative charge or partial negative charge in the center of that ring that can then interact with cations, be those cations amino acid side chains, or sometimes we'll see these with not necessarily in the context of proteins, with other small cations. So a large hydrophobic side chain engaging in a charge-charge interaction with another amino acid side chain. Proline, another tremendously interesting side chain, because it has a cyclic structure. It forms a ring with the backbone of the peptide itself, with its own alpha amino group. This is important because of the structural constraints it places on the polypeptide. In particular, it's interesting that once in the context of a polypeptide, that alpha amino group has no way to be a hydrogen bond donor, and therefore we consider this molecule to be this amino acid side chain to be a helix breaking side chain. More on that later, but for now, we consider that proline is relatively sterically restricted compared to other amino acids. Now, glycine might you might laugh at me including glycine as one of the hydrophobic amino acids. It only has a proton as a side chain, so perhaps it doesn't belong here. But it's interesting to place it here because, unlike proline, which has some interesting steric restrictions because of its cyclic nature, glycine has much fewer steric restrictions, fewer restrictions on rotation around the various bonds of the polypeptide backbone because the side chain is so small. Because of that, we see glycine involved in tight beta turns. As we'll see later, those tight beta turns are structures that allow the polypeptide backbone to reverse direction over the course of only a couple of amino acids. So it allows us to make really close and tight turns. Alanine, another small side chain, simply a methyl group. Valine, an interesting side chain because we introduce branching here at the beta carbon. I think it's a easy mnemonic too to remember that valine is shaped like a V. Isoleucine is also beta branched, but we have an additional methyl group attached to one of those terminal methyl groups. Leucine, of course, also branched, but now at the gamma carbon. So that's our set of hydrophobic amino acid side chains. What we want to think about now is how to develop our familiarity with the amino acid side chains in real contexts. So let's think about what hydrophobicity means in terms of the relative characteristics of these structures. How can we rank them according to hydrophobicity? Well, let's think of that in terms of an experiment called a solvent exchange experiment. And in this case, what we'll do is simply dissolve the hydrophobic side chains in an aqueous environment in water, and then we'll present that aqueous solution with an organic phase and shake it up and see what the redistribution looks like. And of course, we can write that as an equilibrium constant here. K is simply the ratio of the solute that makes it over to the hydrophobic phase, an octanol phase, for example, and related to how much remains in the water phase. Of course, we can write that as a free energy, right? Equilibrium constants are easily converted into the units of energy in this way. We could make those measurements and then spread out the 20 amino acids, comparing their water to octanol transfer free energies, and we see the charged amino acids, whether we're talking about negative charges with glutamic acid or aspartic acid, or positive charges with lysine, arginine, and histidine. We see that those charged amino acids have unfavorable transfer ratios. It's a little bit counterintuitive because all of those unfavorable transfer energies are below the zero line, whereas all of the favorable transfer free energies, those hydrophobic amino acids that prefer the octanol phase, are above the zero line. So we've got that inverted a little bit. But in any case, it's clear to see that the charged side chains cluster with positive free energies of transfer. We can focus on arginine, of course, positively charged with a free energy of transfer of 2. And then if we look over here at the hydrophobic side chains, we see that those all in blue, these aromatics, tryptophan, phenylalanine, tyrosine, all have some of the largest transfer-free energies, most favorable transfer-free energies, which is perfectly sensible to us. One thing that's interesting here that I'm pointing out is that phenylalanine has a significantly greater, almost a kilocalorie per mole, transfer-free energy compared to tyrosine. And again, this arises from the existence of that polar hydroxyl group that's attached to tyrosine. We see the others have a distribution of characteristics. Some are easier to understand, some are more complicated, but leucine and isoleucine and methionine stand out to us as hydrophobic side chains that have large transfer free energies. They prefer to be in a non-aqueous environment. What are we going to use that information for? Once we develop an intuition for the relative characteristics of those side chains, we can start looking at amino acid sequences and predicting their behaviors. This is the amino acid sequence of, it happens to be bovine rhodopsin, I believe, and we see just a string of one-letter amino acid codes. And once we're familiar with those codes, we can start reading this and ranking those, each amino acid in turn, according to its hydrophobicity. And what we readily see, if we map that out, the transfer free energy, for example, if we simply map that out over the sequences, we see a periodic behavior emerging in this particular sequence. I'll just mention as an aside that this is using the Kite and Doolittle scale. There's a variety of ways to score these amino acids according to their hydrophobicity. Not so relevant to us now, simply stated that we can see a periodicity in the hydrophobicity emerging in this sequence. And the prediction that we make then is that there's a periodicity in the structure that would mean that part of the sequence is buried in the core of the protein or otherwise in some hydrophobic environment, and regular elements of the sequence are exposed to solvent. We can look at a protein structure. The same happens to be, again, bovine rhodopsin. This happens to be the three-dimensional structure that corresponds to the one-dimensional sequence I just showed you. And what we see in yellow are hydrophobic amino acid side chains. These happen to be buried in a membrane. So this is a membrane protein structure. These curly alpha helices are all buried in the lipid bilayer of a cell membrane. And so we see again that periodicity allows us, in the characteristics of the amino acids, allows us to intuit or understand the behavior of that protein, predict the behavior of that protein. There's other ways that we can use those characteristics. This is a series of absorbance spectra of, you can see here, the aromatic side chains. I mentioned that earlier that the aromatic side chains, The that large extended pi system gives rise to strong ultraviolet absorbances, and you can see that actually the strength of those absorbances varies dramatically, and indeed that tryptophan, the largest system, has the strongest ultraviolet absorbance. We think of it as having a peak at 275 and a molar absorptivity of 23,000. Respectable. And when we compare tyrosine and phenylalanine, we see that those have significantly reduced not only maxima but also significantly reduced absorbances as well. Phenylalanine being particularly small. And in red here we see a spectrum of bovine serum albumin, a protein for comparison. I'm not sure how many tryptophans bovine serum albumin has, but several for sure. You guys can extra credit if you can tell me in the next class meeting how many tryptophans are in bovine serum albumin. It's also worth pointing out too, though, that the peptide bond has an absorbance as well, and even though the molar absorptivity is significantly smaller than say a tryptophan's, the fact that there are a large number of peptide bonds in every protein means that this absorbance can be quite large. This is a reason that we can't use peptide bond absorption as easily to follow a particular protein, perhaps less unique signal than the tryptophan. So obviously here we can use tryptophan as a monitor of our protein's presence or absence in some cases, and we'll talk more about that later as we discuss protein purification. I want to spend another moment discussing the utility of these characteristics and categories of amino acid side chain characteristics. Let's think about protein purification for a moment and turn away from hydrophobic amino acid side chains to think about how the net charge on a protein might be used to drive a purification process. Thinking about, in particular, ion exchange chromatography, where the chromatographic resin is an ion exchange bead. That is, it has a charge of one type or the other and will therefore adhere the proteins of charged species that are its opposite. So negatively charged proteins will bind to a positively charged bead. Positively charged proteins will bind to a negatively charged bead. The key for us is to realize that we can alter the net charge on a protein by altering the pH of the solution conditions. So at low pH, the basic side chains of the amino acid become protonated and charged. The acidic side chains become protonated as well, but that leaves them uncharged so that the net charge on the protein becomes positive. The converse situation uh, appears at high pH where the acidic residues become deprotonated and charged, while their basic counterparts also are deprotonated but are not charged. Therefore, the net charge becomes positive. So we can select what kind of chromatographic resin we might want to use in terms of ion exchange based on selecting the pH of the solution conditions. As well, we can turn to the characteristics of histidine, which tends to bind a divalent cation, and we can devise a resin that presents a protein solution with a divalent cation. So what I've drawn for you here, or actually a picture I've stolen for you here, is a resin bead that's been decorated with an interesting chemical functional group that allows it to adhere nickel cation. And we see now that if histidines are placed in the appropriate sequence in our amino acid sequence, our protein sequence, that those histidines will interact favorably with that nickel, immobilizing that protein on the resin. So again, we make use of the characteristics of histidine to purify a particular histidine containing protein. Histidine containing protein, that is. Finally, I want to return very briefly to the idea of using hydrophobic side chains to understand protein purification. Let's think about reverse phase chromatography. In this case, we'll adhere proteins of varying hydrophobic characteristics or varying degrees of hydrophobicity to a hydrophobic matrix, and then release those proteins from the matrix by changing the solution conditions, say to more and more hydrophobic conditions, so that under one condition the protein binds. As we increase the hydrophobicity of the solvent, the protein releases. We can make some predictions perhaps of a protein's relative affinity for that sort of a matrix based again on simply scanning the sequence of amino acids and making sense of that sequence in terms of its overall hydrophobicity. So, leaving you with this simple message that we we not only learn
the structures of the amino acids and commit those to memory. We also learn the individual chemical characteristics of all the amino acid side chains so that we can place them into categories. And those categories then allow us to apply our knowledge to real situations in which we might have to make sense of the structures of proteins or the functions of proteins or the behavior of those proteins when we're trying to purify them or otherwise characterize them.